No audio. All right, let's see. Mic, video capture. No audio. Can you hear me now? Mic, video capture. Okay. Can you hear me now? All right, I'm going to delete or turn off my computer. Okay, audio now, good. I think there's a bit of a delay from my end to your end, and part of that is, yeah, I'm running this through an OSB through Logitech through YouTube, so that way I can pull up all these different screens for us, and we can look at the questions you have based on the PowerPoints and things like that. So yeah, so we can see some PowerPoints here. Okay, so I gave you guys a few questions, and so this is for the review session tonight. Basically, what I'm going to do is just answer your questions. So the one kind of ground rule is please don't just cut and paste, a question directly from the study guide. Give me a little explanation as to more of what you're having a hard time understanding. And yeah, I was gonna check and see if there's any email. Sometimes you guys email me questions really quick before. And let's see here, safe zone training. Nope. Let's see, upgrade. Union bookstore. Well, all right. So let's see. What do you guys have? Shoot your questions. Let's go. We'll get this underway. I can tell you that the test will be almost the exact same format as the last one. You're going to have some short answer questions. You're going to have some multiple choice. You're going to have some matching. And on the description here, it's going to be about anything from plant synthesis to plant sex to how plants defend themselves to plant symbiotic relationships. You name it. It's going to be all of that. Okay, so any questions? Let me see if you guys, I got a few of you are zooming in here. Okay, so is a butterfly pollinating different species of flowers not considered a symbiotic relationship because neither species benefits or because it is not considered a close relationship? That is a fantastic question. And, that gets right to the heart of this issue of what makes something symbiotic versus just a plus-plus relationship. Symbiotic relationships, as they are defined, is a prolonged, close relationship between two or more species. And of course, a symbiotic relationship is usually a plus-plus, which would be they are a mutualism where they both benefit. A plus minus, like a excuse me. Plus minus would be like a parasite because parasites usually infect like one host, and then a commensal. Think like some of the like bromeliads growing on the trees in South America. So those are symbiotic relationships. When it comes to a pollinator, pollinating a plant, that is a mutualism. It is plus plus. But if the butterfly is going through all these different plants and just going from butterfly weed to roses to sunflowers and to mints, and it's just pollinating all of these different trees or plants, that is not a symbiosis. That is not a symbiotic relationship because it's not a close, prolonged relationship. But it is mutualistic. Now, that being said, the hammer orchid and the thynid wasp, where the thynid wasp actually goes to that one orchid where the flower mimics the female and the flower is also sending off pheromones. There's a lot of coevolution there. Those two species have been, or at least, they've been evolving together, right? So that would be more on the spectrum, more in line with a symbiotic relationship. Okay, so I hope that explains that answer. And it just goes to show that what we define, where do we draw that line of what's symbiotic versus just a plus-plus relationship? It can be gray in there. Okay, can I explain why diffusion requires energy? Does it mainly involve only passive transport? Oh, that's from, I think, a different class, but that's okay, cool. Diffusion, if you've got the spontaneous movement of a solute down its concentration gradient, right? You put water, if you have a glass, right, and you put some water in the dye, and you don't do anything, the dye will spread out over the entire water, throughout the whole water column until it's equal, reaching equilibrium. The reason why it can do this without you stirring it is because the water has kinetic energy. The air in your room has kinetic energy. You start cooking a pizza, you're going to smell pizza throughout your house because little pizza particles are moving throughout your house. So diffusion relies on the kinetic energy already present in the system. So diffusion absolutely requires energy, and this is an example of passive transport because you don't have to spend any energy to make this diffusion happen. And in our plant class, we have auxin, and auxin will travel through our cells, through plant cells, and it will diffuse down a concentration gradient from one end of the plant cell to another, but then it has special proteins that will help move it across to the adjacent plant cell. So yeah, so there's some active transport and some facilitated diffusion with like the movement of auxin, because it's not random and it's not reaching equilibrium. It's very polar, which means you get more auxin on one side of a stem than the other, because the cells are while it diffuses throughout the cells actually regulate how much it goes in and where it comes into. Okay. How do I figure out how much energy it takes to fix ammonia? That's a good question, and I don't have that on the test, and I don't exactly remember how to remember how to do that. There's a calculation where you have to look at the amount of energy required to break the nitrogen molecule. It's an N2, right? It's got 3 covalent bonds, and you basically have to break that. So you have to, the way we crack it using the Haber-Bosch process is you heat it up, then you react it with a metal like some type of iron compound, and that cracks it, it breaks it, and then you react it with hydrogen gas to form ammonia. I'm not going to ask a question about how much energy that takes. It's well worked out amongst the chemists. Can I explain an example of a symporter? Okay, that's another great question here. Okay, symporters means you've got, let's see here, I don't have an example of it here. We'll have this mutualism here. A symporter is a type of active transport. And you've got primary active transport, think proton pump, where you're pumping protons against their concentration gradient. Then you have secondary active transport. And secondary active transport has a symporter and the antiporter. Okay? Sym means together, like symbiosis. So imagine you're a vacuole and you want to move sugar into the vacuole. No, wait, sorry. Well, that's an antiporter, actually. You're phloem, and you've got a companion cell, and you're going to load sucrose into the Sev tube, right? Okay, this is going to use a symporter. So what happens is you're going to pump through primary active transport. You're going to move protons into the companion cells, all right? You're going to pump them in there. They're out of equilibrium. So what they do is they want to go into the sieve tubes. So there's a symporter, and the protons will go through the symporter back into the sieve tubes. And as they do that, they drag the sucrose with them together, S-Y-M, they drag them together in there. And as a result, you can load the sucrose into the sieve tubes. Again, you can move them against their concentration gradient. So a symporter is using another element, another ion, in this case protons, going down their concentration gradient to pull something with it, it's doing work, against their concentration gradient, and symporters always go together. Unlike an antiporter where they go in the opposite direction, opposites, and and that goes in opposite directions, and that's where you would see it in a vacuole. Okay. Can we go over the sugar transport pathways? Okay, so that was part of the sugar transport pathway. You load it into the phloem, okay? And because you now are loading sucrose into the sieve tubes, well, sucrose is a sugar, It's hydrophilic. It's going to lower water potential. And what will happen is then you will draw water from the xylem into the sieve tubes, and they go under pressure. And you can start to move the sucrose down to where it's called the sinks. And in that case, it's going to now unload the sucrose. It's going to do facilitated diffusion into the companion cells, and then into whatever storage cells are going to do. But then you want to load the sucrose into the vacuoles to store it. Now we're going to go from facilitated diffusion, where you need a protein to allow it to go in there. With facilitated diffusion, you're just going down your concentration gradients using kinetic energy, and then it gets into the vacuole. Now we're going to use the antiporter. Okay? Now in this case, we're going to pump protons using a proton pump. We're going to pump protons into the vacuole. So we're gonna lower the pH in the vacuole, right? We're gonna add all of these protons in there. They're out of equilibrium with the cytoplasm of the cell. They want to go back out. So as they go back out into the cytoplasm, they take and use that energy to snag and pull a sucrose molecule and go the opposite direction. So the protons are going this way, the sucrose molecules are going this way through the antiporter, and then you can do active transport of the sucrose into the vacuole to load it. Taking a couple steps back to the phloem and the xylem, as the sucrose diffuses through facilitated diffusion, it can't cross membranes very easily because it's a large hydrophilic molecule. It's going through these channel proteins or whatever. They're allowing it to move in there. The water potential in the phloem, it goes up because you're removing sucrose. So what happens now, the water will flow back out of the phloem and into the xylem. Okay, so that's kind of how we do the sugar transport there. Oh, I like this one. Selena, what makes regolith different than soil? Yeah, I'm going to plug it. I've got astrobiology on the books for the fall. I'm excited about this class. And I didn't know the answer to regolith and soil. I'm like, why don't you just call it Martian soil? Why are you calling it regolith? It's clearly like this fine material that's not rock. Here's the difference. Soil has a biotic component to it. When we have soil, it's not just broken down rocks. There's also an organic component to it, both from organic molecules and a living component of all of the bacteria, archaeans, protists, fungi that live in that soil as well. So we have soil because we have an active biosphere. Mars and the moon and Venus and Mercury and all these other celestial bodies has regolith. It's just ground up rock and dust with no biotic component to it. Now, that being said, if we go to Mars and we find actually bacteria living in that soil, we're going to have to rethink whether or not we call it regolith or not. And that, we don't know yet. We don't know. We have not disproved the life on Mars. We haven't confirmed it either. I know, we're going down some slippery slopes of philosophy there, but our probes that we've sent there may not be good enough to detect life on Mars. You say I like that stuff. Okay, when plants grow from air, is it because they use oxygen from air? That's a really good question. And actually, the answer is plants do use oxygen from the air, but not because they're growing from it. Let me back up here. Broad picture stuff. Plants are eukaryotes, just like you and me. Plants respire. Plants use oxygen in their mitochondria to make ATP through chemiosmosis. They're breaking down carbohydrates just like you are, and they use oxygen to make carbon dioxide, believe it or not, because they're respiring. Not as much as you are. Not nearly as much. Now, when it comes to this idea of plants growing out of the air, here's what we really mean by that. Plants are mostly, they're like 96, 90, more than that. I forget the exact percentage, way over 90%. Carbon, nitrogen, hydrogen, and oxygen. And it's really about carbon, hydrogen, and oxygen. That's it. I mean, that's really what plants are, right? Okay. I mean, there's other minerals and stuff they need as well, like magnesium and phosphorus and calcium and sodium and things like that. But if you were to take a plant and just break it down to its elements, 97%, 99% or something is carbon, hydrogen, oxygen. So when plants are growing out of the air, you water them, right? That supplies the source of electrons, and it supplies the source of hydrogen for reducing carbon into an organic form, carbon dioxide. So if a plants are taking carbon dioxide out of the atmosphere and they're fixing it into organic material, if a plant is like 90 something percent, excuse me, carbon, hydrogen, and oxygen, that's coming out of the atmosphere with the exception of the hydrogen. And then the nitrogen and the sulfur and the magnesium and all these things are coming out of the soil. And that experiment that they did, hundreds of years ago, the guy grew it like a tree for 5 years, and the tree was, really big, and the soil only lost like a few grams from all of the minerals that it got out of there. So that's what we mean by trees are growing out of the air, mostly because most of their mass comes from the carbon and oxygen, which comes from carbon dioxide, and the hydrogen comes from the water. In fact, when plants are doing photosynthesis, the oxygen that they get from water forms O2 and diffuses out of the leaf. Yeah, some of that oxygen is used in cellular respiration too. So plants respired. It's pretty cool. Okay, in terms of loading a vacuole with sucrose, if the use of the antiporter is the secondary active transport, which part of the loading process is the primary active transport? Maddie, that's a fantastic question. When we're going to go back to this, talking about sugar here, loading sugar into a vacuole requires both active primary transport and active secondary transport. Okay. The primary is you have a proton pump. That is a pump using ATP to move protons against their concentration gradient. That's primary active transport. And by moving those protons into the vacuole, you're lowering the pH, you're making it more positive, you're creating a membrane potential, an electrochemical gradient. That's energy storage. It's like energy stored in a battery. And then the cell or the vacuole uses that stored energy to move sucrose into the vacuole using an antiporter. So as the protons diffuse down, their concentration gradient, they're going to snag and pull a sucrose molecule in there going against their gradient. Okay, go over the acid growth hypothesis. Okay, man, I don't want to redo the whole acid growth hypothesis. What part of that are you having trouble with, Elliot? That's what do you want better explained about the acid growth hypothesis? Okay, there's a fair bit to it. So let me know. Yeah, refine your question a little bit because that's a good question I want to talk about, but I don't want to just like broadly talk about it all at once. Okay, how does SAR work? Well, I think I actually pulled this slide up here. So SAR is a systemic action here, and that is going to be under plants defend themselves. Plants, pollens, plant defenses. And then we can go to here. I'll pull this up. Okay. Systemic acquired resistance, right? So basically what's happening here is that a plant is going to be attacked by a pathogen here. Okay, and what's going on is if you've got a viral pathogen, like a plant virus, and that plant virus is attacking those cells. Well, first of all, you're going to have this other response here. Let me show it to you really quick. Called the hypersensitive response here. And what the hypersensitive response does, this is where you got a virus infecting the cell or a bacteria infecting the cell. So the cells begin to commit suicide, apoptosis. You can see all the brown dots. There's the brown dots right there. That's where those cells are committing suicide, and they might produce toxins, secondary metabolites to target those pathogens. They'll start reinforcing their cell walls, making them thicker. And of course they do the damage. They start doing apoptosis. So if you do programmed cell death, part of what you release during programmed cell death are things like proteases. These are enzymes that cut up proteins. You release nucleases. And nucleases cut up all the DNA, including viral DNA or RNA. Now, because you've got a virus attacking this leaf here, that doesn't mean you want that virus to spread throughout the plant and attack other leaves and other parts of the plant. So now we're going to go to what's called, right here, the SAR response, okay? And what happens is now you've got a damaged cell. And that cell knows it's damaged, so it's going to start releasing cysteamine, okay? And cysteamine is then going to go to these undamaged cells. It's going to bind to a receptor in the membrane. And then it's going to go into that cell, and it's going to start releasing things like jasmonic acid. And jasmonic acid is going to activate things like transcription factors. Think transcription factors, what you're doing there is you're going to start expressing genes. And in this case, you're going to start expressing genes that will protect the plant against that pathogen. So that way it's like an acquired response. Just like when you respond to like a cold virus, I got a cold attacking my body right now. I didn't mean to cough for the defect there, but I have a cold attacking my body, and my acquired immune system is ramping up fighting this viral infection. Plants are doing the same thing. It's a response, and it's ramping it up, and they can also do it to herbivores. So imagine a really good thing with the this active response here, systemic acquired response, is that another way it can activate it is if you're munching on the leaves, right? You got an herbivore munching on it, and the leaves are like damaged, and the leaves will release these signals, and then once again the response is similar. They will start producing these secondary metabolites that might make the leaves taste really bad. So that way the herbivore goes, oh, I can't eat this. It's inhibiting these enzymes in my stomach, so I can't even digest it, so I'm getting sick from eating it. It tastes bad. So the herbivore moves on. And another cool thing about these plant defenses here is that they can volatilize. They can go into the atmosphere, and other plants of different species can pick that up and then start, going through this upregulated response of producing these toxins or these, secondary metabolites that herbivores don't like. Okay. How does auxin play a role in root growth, root growing downward? Man, auxin, the book, I think, does a pretty cool job talking about auxin. Indole-acetic acid, this stuff does a lot of different things in a cell. What it does is it's moving through the cells of the plant root, right? And it's polar. It's not just spreading out rapidly. I mean, it's not just spreading out evenly. It's spreading out based on the plant. If it's in the root, it's going to go to, if it's in the stem, it's going to go to the shady side. That's your acid growth hypothesis. If it's in the root, it's going to cause a root to grow down. So what it's going to do is it's going to inhibit cell growth on the bottom of the root, which is the exact opposite of the stem. Right? The stem it's important with the acid growth elongation hypothesis, but in a root, it inhibits growth. So as these roots on the top keep growing and the ones on the bottom don't, the root grows downward because the cells are smaller here and there's more of them here, so it bends that root downward. And that's the role of auxin in roots growing downward, is that it inhibits growth. Okay, what is one possible theory explaining the origins of sexual reproduction that you presented in class? Yeah, that's a good one. We in our evolution classes, we often talk about evolution of sexual reproduction because it maintains genetic variability in a population, which allows a population to rapidly respond to a changing environment. So it's a maintenance of genetic diversity. Whereas it doesn't actually create genetic diversity, that's mutations, but it's like a deck of cards, and all the cards represent different combinations of genes, and some combinations are winners and some combinations are losers. But what we rarely talk about is the origins of sexual reproduction. And the theory is the mitochondria did it theory, I guess. There's probably a more scientific name for that. But basically, here's how it works. Sexual reproduction is defined as combining two genomes from two different parents to form one new organism. That's a bit black and white. Talk to some fungus, and there's some different mating types there, but basically that's what you're doing. Think about plants and animals. We have male gametes, female gametes. They combine. They're haploid. They combine to form a diploid organism. That's sexual reproduction. And the reason why we think that it evolved is because of mitochondria residing inside of these cells, and mitochondria using oxygen create reactive oxygen species, ROSs. And what those RSs do is they damage the cells. They can oxidize membranes. They can oxidize proteins. They can damage your DNA. And if you damage a crucial DNA gene, a crucial gene, that cell could die. All the mitochondria in that cell die with it. So the evolution of sexual reproduction was, if we could have two copies of a genome, then if one gene becomes damaged, you've got a backup. And in fact, even today, many of us carry deleterious alleles. These are alleles that are mutations that don't function well. You don't know because you're heterozygous. You have a functioning copy. Like a mutation that knocked out A blood is O blood. That's a mutation. O is a mutated A. It doesn't produce the whole antigen, right? Color blindness. Guys, if we get the gene, we're colorblind because it's on the X chromosome. We only get one. Ladies are rarely colorblind because they would need two copies. They would have to have basically a mom that's a colorblind carrier or colorblind and a dad that is colorblind. So the idea here is that with sexual reproduction, that the mitochondria did that so they could bring in a backup copy of their genome, so if the cell dies from damage they caused, basically, they don't go down with it. So that's a good one. Okay, Elliot, excellent question, man. How are the expansins activated? Okay, so you got this. You got a plant bending toward light, man. You could be walking around in the woods and you see the branches grow into it. You could see something like a sunflower moving across. This in the morning it's pointed toward the east, and in the evening it moves and points toward the west. And it's this acid growth hypothesis that basically has these expansins in it. And here's how it works. Plant cells are surrounded by a cell wall. And that cell wall is made up of cellulose microfibrils. And one of the ways that makes those microfibrils very, very strong is a cross-linking in a lot of hydrogen bonds. Hydrogen bonds are weak, but so is an individual string. But yet, put thousands of them together and you have clothes that are pretty strong. Lots of hydrogen bonds, put them together, they're strong. So what happens is, as a plant is going to grow, is going to grow long, what the plant has to do is relax those microfibrils. The cellulose microfibrils. And the way it does that is by using an enzyme called expansins that basically act like little scissors cutting up those hydrogen bonds, which relaxes the cell wall. So what it does is the cell won't get wider, so to say, but it will elongate this way. It's like taking a balloon and wrapping string around it and then relaxing the string so the balloon can expand like lengthwise. And the way those expansins are activated is you've got a receptor on the tips of the branch called the phototropins, and they activate auxin, and auxin goes down the shady side of the plant. So there's your polar. There's more auxin on the shade side than on the sun side. And what they do, the auxin will bind to a proton pump and turn it on. So here's our proton pumps once again, and we start pumping protons right outside the cell membrane into the space from the cell membrane into those the cellulose, into the cell wall. And it's called the acid growth hypothesis because you're acidifying that region by pumping in those protons. And those protons activate the expansins, which then cut up all of start cutting the hydrogen bonds, allowing the plant to elongate. So good question. Okay. When distinguishing between pathogen and the herbivore, are you looking at where it is affecting the plant, like on cellular physiological sense for a pathogen? How do you know it is an herbivore? Wow, Melissa, you did a really good job there. You basically halfway answered your question. You're right. A pathogen, the way we're going to talk about it in this class, you're basically disrupting the physiology of the plant right down at the cellular level. So if you're a virus and you invade a plant cell and you take over that plant cell to make more viruses, you're affecting that cell's ability to function inside the plant. You're affecting its physiology. Different viruses require different proteins to attach to that cell and invade that cell. So viruses might attack phloem cells, like companion cells. Viruses might attack mesophyll cells. Viruses might attack, other cells of the ground tissue. You get the idea. So it's affecting the physiology. An herbivore eats plants. They're going to come along and munch the leaves. You might even have these little tiny moths. They're called leaf miners. They will actually get inside of a leaf and eat the leaf inside the epidermis. So they're pretty sheltered from predators like that. You can go see those in aspen trees. You can see all the little white tracks on the underside. But lots of plants have leaf miners. They're not really affecting it at the physiological level because they're still just munching on the leaf. When the stomata open, do the phototropins signal for the synthesis of auxin like they do for cell elongation? Ooh, Maddie, that's a good question. And the answer is basically yes, because what happens is when you have blue light in the morning, the sun is coming up, light is starting to get going here. Those stomata need to open up so you can start doing photosynthesis. So yeah, they're going to start sending those chemical signals down there and cause those cells to open up. So yeah. The phototropin signal for the synthesis of auxin. In some plants they definitely will, because plants are going to definitely grow to the light. So that's a good question. So yeah, they're gonna signal for elongation as well. That auxin and all these hormones, they have so many different purposes inside our cells. Like ABA is another one. We talked about ABA being sent up from the roots when there's not enough water to close the stomata. But ABA with another one is a gibberellins, which also affect like plant growth as well by inhibiting it, and the gibberellins are like increasing it. So there's all these different hormones with all these different things. The explanation sounds like a cell-mediated response. Yep, some of these are definitely cell-mediated responses for certain. All right, you guys, some fantastic questions right here. Do you have any other questions about this test? Fantastic. It's really fun hanging out with you guys, even though it's kind of a one way, but I love the top chat here. I love answering your questions. Good luck on this test. I will tell you that the test, like the last one, the study guide, the questions will be pulled off the test just like this last time. So, if you know this stuff well, you won't have any surprises on the test. It's exactly what we've talked about in class and on that study guide. All right, any other questions? Oh, antipodal cells? Yeah, okay. In plant sex, basically, anti means like opposite, right? So you've got them, if you think of let's see if I have my image of a flower up here, right here. I can show you. You've got the micropyle down at that's at the bottom. And the antipodes are above the, let's see here. Yeah, why? Oh, it's over here. So let me just show you this slide right here. Okay. So if you look at this micropyle down here, right? At that micropyle, this is where the egg is. All right. And this is at the bottom. This is the ovule. This will turn into the seed. And up here, these are the antipodal cells. Basically, what these antipodal cells are going to do is they're going to help nurse a growing embryo of the plant. That's their main role. Okay. Some symbiotic, a symbolic relationship that is for defense. Yeah, there are symbiotic relationships for defenses. Going back, we're going to switching out of plant life cycles here. Yeah, so a really good example of a plant symbiotic relationship for defense would be the plant that releases the pheromone to attract those parasitoid wasps. And once again, I would label that a symbiotic relationship because there has to be the coevolution where the plant is evolving a chemical hormone that is close enough to the actual hormone of the wasp to respond to that very specific chemical to come and lay the eggs on the caterpillars just munching on it. So I would say that's symbiotic relationship because there's coevolution going on. Double fertilization and why this is important. Yeah, so double fertilization, let's go to, I got the slide up for that right here. On double fertilization, if you notice in plants, you've got the pollen grain, and the pollen grain has basically a generative cell, which is, through mitosis forms 2 sperm cells. And then it's got a tube cell. And the tube cell actually grows a tube down the stigma, right? And then it comes down here like this, and then discharges the 2 sperm into the ovule here. And here's the egg. One is going to fertilize the egg. That's one fertilization. The egg becomes diploid. The second sperm is going to fertilize these polar nuclei, and then other images, other diagrams, you might see this polar nuclei as 2 cells. Either way, the sperm cell is going to fertilize this, and this will become the endosperm, and the endosperm is triploid. Okay, so that is the double fertilization. Like, unlike an animal, when there's a fertilization event, it's just the egg meets the sperm meets the egg, and that's it. There's nothing else going on. Here, 2 things are getting, are forming recombinant DNA, basically. So, pretty cool. Plants mostly grow out of the air. Explain the reasoning behind this statement. Can you explain this question? Hey, Matt J23, you'll back up a little bit on this one. I went over that a few minutes ago, but basically the importance of plants growing out of thin air, out of the air, is that the majority of a plant's mass is carbon, oxygen, and hydrogen. It's like over 90%. The hydrogen comes from water from the ground as you split the water, but all the carbon and oxygen of a plant comes from the carbon dioxide out of the atmosphere. That's what that means. Okay, the length of time a plant is exposed to a certain wavelength of light can inhibit or promote flowering. What is the far red light mimicking? Okay, think about a plant. You want to germinate. Okay, so this is, you got some photoperiodism going on here, or photoresponse. So here's what happens. Flowering, sorry. Okay, so in plants have different flowering strategies. Some plants bloom all year long. Some plants bloom in the spring, some in the summer, some in the fall. So plants are sensing environmental cues of when to start expressing all the genes to flower. Okay? So if you bloom in the summer, you're a long day plant. If you bloom in the spring or fall, you're a short day plant. Now, there's more going on to it, so they can differentiate between fall and spring. But basically, when you are mimicking far red light, you're mimicking like shady conditions. So imagine in the spring, in the winter time, the sun is lower, the shadows are longer, the wavelengths of light are longer. But a short day plant would see that and go, "Hey, it's a short day. I'm going to bloom." Whereas if you're a long day plant and you see that far red light, 720 nanometers, you're like, wait, it's a long day. I mean, sorry, it's a long night. It's a short day. I'm going to inhibit growth. All right. But if you switch it up and hit them with shorter wavelengths of light, like 670 nanometers, that will induce a long day plant to grow, but will inhibit a short day plant. Yeah, Bob, outline the main steps of embryogenesis. Hey, one of my criteria here was not to just cut and paste the question from the from the study guide into this, like what part of embryogenesis are you having a difficult time with? And so if you have a specific question about embryogenesis, yeah, let me know. And same thing goes with the ABC model of gene control over gene structure. And actually, as I am putting the test together, I'm not gonna have anything on there on the ABC model. Actually, I'm gonna talk more about the embryogenesis, but I do want to get a little bit into GMOs tomorrow and have a little bit of time to talk about that. Melissa, can you touch on the consequences of the Haber-Bosch process? Is it just about breaking down into and making ammonia, or looking at the downsides? Yeah, so there's consequences to it, right? The Haber-Bosch process has done several things, both beneficial and at a cost to the environment. One of the benefits is like our ability to grow crops is crazy. I mean, we would have to feed 8 billion people. We would have to use 4 times the amount of land that we currently use to grow crops if we didn't have the Haber-Bosch process. So it's one of the reasons why our crop yields are so so dense, or so good, because we can do a lot. We can grow a lot more per acre than we used to, because we can add the nutrients the plants need. But by doing the Haber-Bosch process, we have basically doubled the amount of nitrogen into our ecosystems. And how that's affecting the relationships between, like, plants and their mycorrhizal fungi, because we know that can switch from symbiotic to perhaps not symbiotic, more parasitism. I don't even know how well that's been studied, but for me, when I studied my PhD, I was very interested in aquatic ecosystems, and nitrogen in aquatic ecosystems. I mean, that's like rocket fuel for algae growth. And so aquatic ecosystems are incredibly sensitive to nitrogen. So with all the fertilizer we use, not all of it is used by the plants. Lots of it gets washed away into our streams and rivers, which increases algae growth, and in our lakes it causes eutrophic zones. And then the Mississippi is huge. It drains most of the Midwest, right where we farm, and it drains right into the Gulf of Mexico. And there's so many nutrients that it just causes these intense algal blooms. And as the algae die, they suck up all the oxygen, because to break down organic material requires oxygen to do it efficiently. So it creates these anoxic zones that create, fish kills. So that's one of the big problems there. So that's some of the downsides to it. Do embryogenesis have fruits and seeds? Well, hey, so the embryogenesis, what's going on there? And I was just working on that slide here for tomorrow. I believe I have it here. No, that's the wrong one. Oh, I didn't click on the... Aha! So the thing with embryogenesis here, basically the whole idea of that is, this is how, when you go from that egg, how do you grow into a little embryo inside the seed? So we're not going to have the fruits. It's just about how do you grow the little embryo inside the seed. And there's some cool steps. The book does a fantastic job of outlining it. That's why I'm like, hey, on page 528, what you do is once your zygote is fertilized, what happens is it immediately undergoes mitosis, right? And you get, a basal cell and an apical cell, so there's already this uneven, distribution here. And the basal cell, as it starts to grow, the apical cell starts to grow into a heart-shaped box. I'm thinking Nirvana. It grows into a globular heart-shaped cluster. We start forming the primary axis of the body plan of these plants. And once you start getting to a globular cluster, and one of the most important things about embryogenesis, much like us, that as those cells begin to differentiate, because when they're early on, they're not differentiated cells, they will then start to establish this primary body plan, the lengthways and then outward, which is the radial. Basically, as they start to grow and differentiate, they do so based on their location. So the outside cells, those are going to go off and form the protoderm that will form the dermal tissue. Just inside of the protoderm are things like the ground meristome. Those cells will differentiate into those crazy sounding cells, the parenchyma, the sclerenchyma, and the collenchyma cells. But the ground tissue, like, think of mesophyll cells, the parenchyma cells, right? That's where photosynthesis is occurring. And then in the middle of the plant, or this middle of the embryo, going up and down it, you'll start getting the procambium cells. And they will begin to differentiate into the vascular tissue, the xylem and the phloem. And of course, xylem is made up of a couple different types of cells. The phloem is made up of the sieve tubes and the companion cells. So that's what's important with the embryogenesis, is going from that fertilized zygote and then how it starts to divide, one on top of the other, right? And then it forms a globular cluster that's like a heart. I'm not making a good heart with my... Oh, man. I'm horrible at this. Can't wait to get my chalkboard up so I can do this. But at any rate, that globular cluster, then those cells begin to differentiate. Man, that point, I got the point right here. Man, plant cells differentiate based on where they are in the body. And that's important for animals too. Although one big difference that we might learn about in the plant and the animal part is animal cells, they're in embryology, move. They move around, which is kind of cool. Okay. That question was asked in class. Can virus change plant DNA and cause cancer? And the answer to that is I really don't know. And you're the second person to ask that. And I'm gonna ask around and see if I can look some stuff up. I suspect that cancer is basically a price to pay for being a multicellular organism. Now, the difference with a plant is if a leaf gets cancer, it can just kill the leaves and just die of apoptosis, right? Whereas if a human gets it and it spreads, we can't live without an organ. We can't live without our kidneys. We can't live without our liver or our stomach. So as it spreads, it kills us because our organs shut down. Whereas a plant, well, if you get some crazy growth over there in that leaf, just have the leaf commit apoptosis and be done with it, and you never see it. So I imagine that they do get it, but because they can just kill everything around it. I'm just guessing here that it might not be a problem for the plant, but I really don't know. I'm just guessing here. I'm speculating. Okay, on the study guide, when you ask us to know ecological interactions, are you referring to things like mutualisms, parasitisms, commensalism? Yeah, right off the slide. Yep, Maddie, that's right off the slide there. Just to remind us of some ecology. Because one of the things that with plant and animal form and function that I try to remind us is that, form and function is really a lot about physiology and how an organism is adapted to their environment and responds to their environment. We need to know our cell and molecular biology. We need to know about genetics in terms of like how genes are responding. And we need to know the ecology. What are the conditions, these day-to-day interactions our plants and animals are dealing with. And then the evolution, because the day-to-day interactions drive evolution and the adaptations that they have. And so mutualisms, parasitisms, commensalisms, those evolve over time based on these day-to-day interactions, and they affect these things right down at the cell molecular level and at the genetic level. Okay, so we got the primary functions for polar nuclei, eggs, synergids, and micropiles. So let's go back to our plant here. And then let me show you. That's the endoderm. And then we can go right up to here. And we can see on this slide right here, I pretty much kind of put some of this stuff in there, right? What these things are important for. So, the egg is pretty obvious. I mean, this is gonna form the embryo, right? I mean, you retracted. Oh, I got the slide right here, but it's a good question. The egg gets fertilized, and here's something that you might want to know. What are these little synergids doing right there? What's the purpose of those? And basically, when the sperm cell enters through the micropyle, those synergids help guide it and then close it off after there's been a fertilization. So that's kind of cool. And then the antipodes, I got it right there on slide, I believe. I think it's got a number on it. 32. Yeah, and then this, the synergists stop pollen growth. The antipodals are important for nutrition. So, yeah. All right. Any other questions here? Are we all good? All right. Well, I will see you guys tomorrow. We will talk a little bit about this embryogenesis and a little bit about pollination syndromes, which is kind of cool because that's how these plants are responding to the environment by attracting different pollinators. And then we will talk a little bit about GMOs because it's highly controversial, but maybe not as much as it should be. Is there any extra credit? No, no. The class average last time was pretty good, actually. You guys are doing well. And then last summer when I taught this class, the class average actually came up like, almost 10 points from between test 2 and up to test 3. So uh, my biggest suggestion to you are answer these questions, answer them thoroughly, work in groups. You answer those questions as best you can on your own. Do most of the groundwork yourself first. Form study groups like tonight, tomorrow night, Wednesday night. And when you do your study groups, make sure that you guys all have similar answers, and that if you don't understand something, when they teach it to you, you teach it back. Don't just say, "Oh, I got this," because you might not. So teach it back. And yeah, the book is a great resource. This is a good resource. I've got some YouTube videos, podcasts, and then you can use the Wikipedia too if you want. Oh, I wish I had taken this class with you. Oh man, yeah. Well, we'll see if I'm going to teach it again this summer for the last time. We'll see. I don't know. We're having some changes up in our curriculum here. Okay.